Hello, everyone. Welcome to today's live broadcast, PCR Testing in Rodent Health Monitoring Program. Where are we exactly? I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by the Andersons Lab bedding products. As the sole manufacturer of bed coves, the Andersons has provided the research community with the most trusted name in bedding since 1962. From irradiated and qualified products available in autoclavable and bulk bags to sterility and heavy metal testing, the Andersons provide a number of services to the industry. For more information, please visit www.andersonslabbedding.com. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the green Q&A button lower left. I would now like to introduce today's speaker, Dr. Mathias Leblanc. Dr. Leblanc is the Assistant Director of the Animal Resources Department at the Salk Institute for Biological Studies. He obtained his doctorate in veterinary medicine from the University of Montreal in 1997. Soon after, he pursued a master's and a PhD degree in physiology and endocrinology. Dr. Leblanc did a residency in veterinary pathology at the University of Montreal while remaining active in lab animal science. He moved on to the position of Assistant Director and Veterinary Pathologist at the Salk Institute in 2007, where he is responsible for the veterinary care, diagnostic, and preventative medicine program, as well as the comparative pathology program. Dr. Leblanc is also a staff scientist in the Gene Expression Laboratory and has published on cancer research and metabolism in several high-impact journals such as Cell, Cancer Cell, and Nature Medicine. I will now turn it over to Dr. Leblanc for his presentation.
Thank you, Judy, and good morning, everybody. I hope everybody can hear me well. As Judy said, I'm going to talk about environmental PCR testing and rodent health monitoring. Where are we exactly? Just want to say a few things. First, I just came back from a Europe vacations in Europe, in Paris, Spain, and Portugal. So I've added some pictures of my trip in the presentation. So if you get bored, well, at least I hope you're going to have a good time looking at the pictures. So I'd like to talk about environmental PCR and testing. I'd like to start with a very simple definition. What is doubt? Doubt is a noun, and based on the Webster Dictionary, it means a feeling of uncertainty or lack of conviction. And why did I start with that definition? It's just to say that health monitoring in general is not an exact science. It's very complicated, just as also diagnostic assays. We think that, everything is foolproof in this area, and that we should not doubt what we have. When a lab or a given lab gives us results, we should think, well, you know what? If the lab's found a positive, it's positive, and that's good. Well, that's not true. As in science, you should always doubt and remain critical. Remain critical and ask questions. That's the best way to go. And I hope that this presentation will prove that point. Like Leonardo da Vinci said in French, he said, "Qui ne doute pas acquiert peu," which means who do not doubt acquires little. You need to doubt when it comes to health monitoring and diagnostic assays. Always doubt a little, and don't be afraid to ask questions. So soil bedding sentinels are definitely not always efficient and reliable at revealing the presence of pathogens in mouse rodent colonies, especially when they're housed in individually ventilated cages and when the pathogen prevalence is low, which is the case for most of your facilities. With the advent of new molecular techniques, several investigators and several veterinarians have proposed alternatives to soil bedding sentinels, such as PCR analysis of environmental samples from either the cage or from rack exhaust plenum to improve the detection of pathogens. And that's great. And I believe that we're going in that direction. So taking advantage of the sensitivity of PCR assays, different investigators have suggested to sample areas where agent-laden particles accumulate in high concentrations, such as exhaust filters or plenum of ventilated racks, to detect pathogens at the rack level. There was some original success, especially with aerosolized viruses like Sendai or MHV, and definitely with termites. There was a good publication on that. But then some have gotten very excited and things got a little bit out of hand. I got this quote from a publication that was given to me by my colleague, John Reuter, who was the senior director of the Animal Resource Department. Gave me that publication a few months ago. And when I read it, I was kind of flabbergasted. I was like, what is this? And the publication said, using swab of plenum or rack HEPA filters to monitor a rack for infectious agent completely eliminates the needs for bedding sentinels. For exhaust air dust compatible racks, we can detect virtually all agents by air sampling, many more than with soil bedding sentinel. Well, I do understand that soil bedding sentinels is not perfect, but I went back to my colleague, John, and I said, where did they take that? Did I miss something? I didn't read all the publications or all the literature? And he said, well, I don't know. I was as surprised as you. The fact is that there are very few systematic studies on the efficiency of exhaust plenum to detect the presence of pathogens in naturally infected, and I do say naturally infected colonies, or studies that have determined the optimal environmental sampling strategies for those exhaust plenum PCR for different microorganisms. So when you read things in publications, always stay critical and say, well, is that true? Is that absolutely true? When I read this, I said, no, that's not possible. And I went back, and I hope that this case study, that we had here at the Salk Institute will try to illustrate that well, and illustrate that health monitoring in general is not straightforward, and that whenever using new technologies or new testing paradigms, you need to remain critical, ask questions, and remember some basic concepts, especially before taking costly action that may be disruptive to your clients, namely your research community. Always interpret results very cautiously with an overall understanding of this testing methodology, the intrinsic controls, and all the variables that may affect the results. Just as in any diagnostic test, PCRs are not bulletproof, and don't take for cash everything you read in publications. So let's talk about the Salk Institute. I'm going to talk about our program just very quickly, so to give you an overall context. The Salk Institute for Biological Studies is in La Jolla in the San Diego region. We're a nonprofit research institution that was funded by Jonas Salk in 1960, the inventor of the polio vaccine. We consistently rank amongst the leading research institutions in the world. In fact, our institute has trained about 2,700 scientists, including 5 Nobel Prize winners, and our labs are currently home to 9 HHMI investigators and 14 members of the National Academy of Science. Our major areas of research is molecular biology, genetics, neuroscience, plant biology. We have a typical medium-sized animal care news program. The Salk Animal Resource Department supports the teaching and research of about 47 faculty or labs and their students. We are, of course, ALAC accredited, and we operate about 45,000 square feet of vivarium, including 2 mouse barrier facilities, namely SAF and TG. The names are important later to understand the presentation. A conventional multi-species facility, CRAF, and 5 other satellite facilities. Most of our rodents are housed in the barrier facility, SAF, with an average daily census of about 14,000 cages, mostly in micro-isolators on ventilated racks. Our biosecurity program is fairly standard. I just want to remind what is a biosecurity program. It's all the measures taken to prevent, contain, and eradicate pathogens. So our Biosecurity program is standardized in all our facilities and is regularly optimized according to contemporary standards to minimize pathogen outbreaks. Of course, being an academic institution, there's a lot of challenges, a lot of investigators, lots of postdocs that do not necessarily follow procedures. So we do have breaches in our biosecurity program that leads to outbreak. But nonetheless, we have a very stringent biosecurity program. For example, we have restricted access to our facilities with a stringent training program. All our investigators, postdocs, must go through annual retraining when they work in our facilities to make sure that they follow procedures and are up to date. We have a highly regulated traffic flow for both the staff and the animals, with a one-way out traffic flow for mice in our barrier facility. Once they leave the facility, they never come back. All transfers of animals between facilities and rooms must be approved, handled, and documented by the Animal Resource Department. And that's very important for us because whenever there's an outbreak, with the movement of animals, we need to be able to look at where the animals from positive rooms have gone so that we can trace back and risk assess the outbreak in other rooms. We have strict PPE. All our imports from non-approved vendors in the barrier must be embryo re-derived and tested prior to release. All our biological are tested for murine pathogens prior to use. That's only a few components of our biosecurity program. But as I said, in an academic institution, as most of you know, it's very hard to maintain this program, and breaches do occur, and we do get pathogens outbreak, and that's why we do have a health surveillance monitoring program. Our health surveillance program is meant to give us a broad overview of the health status of our rodent colonies. But always remember that these programs, especially those based on soil bedding sentinels, they're not efficient at detecting all pathogens in rodents. And also, results are retrospective. So whenever you have results from a health surveillance program, remember what you have at that time and point when you tested the animals might have been there for weeks to months. So take your time to analyze the result, understand and confirm the result, and take your time before taking any harsh decisions. It's not because you have a positive that you need to act immediately. So our health monitoring program is based on soil bedding sentinels. I'm having trouble with my computer, I'm sorry. So we have, it's fairly standard, 1 sentinel cage per half rack that contain 2 naive wingling Swiss Western females that are replaced and tested serologically every quarter for pathogens. And then once a year, all our sentinels are also shipped to a reference lab for comprehensive necropsy, bacteriology, fecal PCR for endoparasites, and PEL PCR for ectoparasites. Then any positive results must be confirmed on the sentinel cohort by a secondary method, and as well as on colony mice before we do anything. So a bit more about our mouse barrier facility. The vast majority of our mice housed in our barrier facility are on microisolators on Allentown HEPA filter racks, and all the mice must be manipulated in ATS, animal transfer station, or BSL-2 cabinets using strict aseptic technique with liberal use of Verconess disinfectant. A few of our mice are housed on static microisolators and handled in open air in our behavioral testing suite. Our mice are fed irradiated rodent chow, are given RO water, and cages have corncob bedding and crinkle nests for enrichment. The irradiated rodent diet, I just want to mention one thing. We had MPV here at the SOC for about 15 years, and we were convinced, based on the epidemiology of our outbreaks, that it came from the feed, and we switched from irradiated diet about 5 years ago, when we completely eliminated MPV from our facilities. Another point is that in our mouse barrier facility, and that's an important point, racks are disassembled, fully decon, and autoclaved annually. Our cages are on a 7 to 10 days cage changing cycle. Most cages, the vast majority of our cages In our non-autoclave rooms in our barrier facility are sanitized in our tunnel washer with standard 180 Fahrenheit water. But about 5% of the cages are autoclaved with the bedding and the crinkle nest for prior to use for our immunodeficient and sensitive strains. So to start the case report, we have to come back to June 2013. That was our big sentinel quarter where we sent all our sentinels out to our reference lab for necropsy, etc. And we were waiting for the results of the sentinels. Everything had been sent, and I remember very well. It was raining that day, and I was coming to work. That's very rare in California. And in San Diego, I think it's rained what 5 or 6 times since I've started in almost 7 years or 8 years I've been here. And so I sit down on my email in the morning, and I get a dreaded email saying, well, there you have positive you have positive results. So I go see, and I see that 2 sentinels in 2 different rooms in our barrier facility staff was positive for Spicularis tetraptera on fecal PCR. Good. Well, that's not a good news. Pinworm is certainly not something you want in your facility. And we had firmite outbreak in the past, but we didn't have pinworm outbreak. So we're kind of not very happy with it. But we said, we need to confirm that. It's an unexpected positive result. Let's take the time to confirm that. So we did secondary confirmation. We did fecal flows and direct fecal exam on the sentinel. We asked the reference lab to do that. And then we had the cage cohorts of those sentinels here still at the institute. So we did that, and we also tested colony animals. And you know what? They were positive. We saw some worms in the cecum. We truly, really were confident that those 2 rooms were positive. All the other rooms in our barrier facility and all our facilities were negative based on fecal PCR. So at that point, we say, is it a contained outbreak or isolated outbreak in 2 rooms, or is there something more? So the first step was to quarantine the room, implement strict traffic flow, and confirm the results. And that's what we did. Again, always consider that sentinel results are retrospective. So it's been several weeks to several months since it's been there. Always take time to confirm and think about the most appropriate plan before taking harsh decisions that may be costly and especially get on the nerve of your scientific community. Once you've confirmed your results, well, you need to determine the extent. You always need to chase. You don't want to chase your tail, especially with outbreaks. You might have pathogens in 2 rooms by sentinels, but it doesn't mean that the pathogen is not present in other rooms, especially in an academic institution where you have so much collaborative work. It's very probable that your sentinels have not picked up a pathogen in another room. So you need to determine the extent. Once you've determined the extent, you know what you have in your facility, then you need to communicate to your investigator, then treat and test the efficiency of your treatments. And I want to touch base quickly on communication here. Communication for us is absolutely essential when you have any outbreak. It's essential for the success of our pathogen containment and eradication plan, and if you don't communicate effectively with your scientific community, you're going to have problems. You need to reach out to them. You need to organize meetings, explain to them, and make them feel that they have a vested interest in resolving the situation, and that they will play an active role in resolving the situation. We do open conferences. We do meetings with lab managers. We do emails and written communication, whatever. In general, I can tell you that in written communications are very poor at conveying a message. We always get, people don't read their emails. People don't read letters. However, we find that lab managers are the best people to work with. The lab managers in general are the bridge between the PI who was too busy to take care of problems at the facility level and the postdocs that are too busy with their research. If we can keep the lab managers involved and make them feel that they have an important role in our program, that's an amazing step in reaching a successful resolution and managing successfully any pathogen outbreak. So considering the biology of the parasites, of the pinworms, its persistence and resistance in the environment, and the collaborative nature of research in academic institution, we were very concerned that the 2 isolated cases was just the tip of the iceberg, and that the outbreak also involved other rooms. As such, we said, well, we need to implement a very large testing program in all facilities to determine the extent of the infection before deciding on a definitive containment and eradication plan. So how did we do that? First, all the remaining sentinel cohorts that we still had were tested by fecal floats, perianal tape test, because sometimes you have dual infection with Aspicularis and Cephassia, and direct cecal exams. All were negative. Good. So basically, our fecal PCR were negative in all the other rooms, and now fecal floats, perianal tape test, and cecal exams were negative. Gave us some confidence that it was isolated, but that's not enough. We also tested all the transferred animals from the positive rooms to other rooms in the past year, as well as their offsprings or relatives still present, by fecal floats, perianal tape test, and when possible, when the labs were willing to give us animals, by direct cecal exam. This was possible because we track all transfer between rooms and facilities. And I think that's very important in any pathogen outbreak management. All those were negative. Again, so it seems that there were no animals that it looked like that, at least to the best of our knowledge, there were no animals that were transferred to other rooms were positive. Then we did environmental PCR testing on room surfaces and facility HVAC in both the 2 positive rooms, the 2 confirmed positive rooms, as well as the behavioral and procedural testing suite, where the mice of positive rooms could have been used in open air. That's because it's well known in rodent colonies infected with pinworm that are conventionally housed, and when you do open air, you can contaminate your HVAC, you can contaminate surfaces. So we thought, environmental PCR testing of room surfaces and HVAC might be a good way to see if we have contamination, environmental contamination. All were negative. Wow, surprising. Seems that everything is in those 2 rooms and it's well contained. At that point, we were not satisfied. That's where where problems start. We said there was a lot of talks based on several publications that we could test at the rack levels. We could test plenums of rack and determine if an agent is there at the rack level by testing ventilated racks. And most positive pressure ventilated racks, including our Allentown racks, operate in similar fashion. Basically, you have HEPA filtered air that is circulated in cages at 60 to 70 air changes per hour, which creates the barrier at the cage level and also eliminates or reduces the waste products from the cage. Then air from each shelf is collected and channeled through an horizontal exhaust manifold, and then to a common vertical exhaust plenum before being exhausted in the room or the central ventilation system based on how it's hooked up after the air has been HEPA filtered. So several investigators have suggested that we could take advantage of the sensitivity of the PCR assay to sample areas where agent-laden particles accumulate in high concentration, such as the exhaust plenum of ventilator racks, to detect pathogens. The strategy was proven useful and very sensitive to detect termites at the rack level. But at the time, there had been no publication on the efficiency of that testing strategy for pinworm. Nonetheless, we said, you know what? Pinworm eggs survive in the environments for weeks. Eggs were found in the past in the environment of conventionally housed and infected rodent colonies. And although Aspicularis eggs do not aerosolize as easily as Cephassia, we said, well, you know what? PCR might be sensitive enough to detect only a few eggs or just residual genetic material in the environment in those plenums. So we decided to take a leap of faith and be a bit of and be explorers. And we asked our question. John and I said, can PCR on rack exhaust plenums be a reliable and sensitive method to assess the presence of pinworm DNA or infection at the rack level? And we tested all our racks by pool PCR in all our rooms, in all our facilities. And that's when we had our big surprise. So let's go look at this table. In red, what you have are the SAF44 and SAF50. Those are the two confirmed positive rooms. You have the PCR, fecal PCR that was positive, the fecal float that were positive, and as expected, not surprisingly, the exhaust plenum PCR were positive. Great. But then, we should not, all our sentinels in all the other rooms were negative. The floats were all negative. The tapes were all negative in all the other rooms. And yet, big surprise, all our plenum PCR were positive. The vast majority of our racks in all our facilities were positive. All except in the rooms, in the autoclave rooms, where the cages are with the corncob bedding and the critical nest are autoclaved prior to use. So that was extremely, extremely disturbing for us. We said, oh my gosh, we have a widespread infection in all our facility, but why didn't our sentinels pick it up? You know what? Pinworms don't necessarily, well, they transfer fairly well, but they, we don't necessarily test our sentinels very well. It's not very sensitive in our sentinels. So we might have missed it in our sentinels, so we were very concerned, but we had to determine what was going on. So we had three possible explanations to explain those results. First, a widespread outbreak of pinworm in our facility that selectively affected non-autoclaved rooms. Or, second thing is that what we had was not a true pinworm infection in all the rooms, but what we had was a historical presence of pinworm DNA in the exhaust manifold without active infection. That's possible. Or just our third possibility was that the false positive environmental PCR results was due to non-specific amplification of DNA. So we had to exclude all the three. We had to work on the three hypotheses, one after the other. A widespread outbreak that selectively affected non-autoclave rooms would have been very surprising considering the negative sentinel results in our biosecurity program. Nonetheless, we decided to exclude that hypothesis by testing index high-risk colony animals from representative autoclave rooms and non-autoclave rooms. So basically, we tested the autoclave rooms that were negative by plenum PCR and the non-autoclave rooms that were positive by plenum PCR, and we chose the non-autoclave rooms with the lowest crossing point value on the plenum PCR, which means that they have the strongest PCR signal. We wanted to increase our chance of finding it in mice. And what we did is we tested breeding and young colony mice, basically 5 to 10 week old colony mice, and we decided to test those groups, those animals specifically because they are the ones that have the most pinworms in general. That's the best. Those are the best candidates to find it. So we did on those animals in the representative autoclave rooms and non-autoclave rooms. We did fecal floats and pooled cage PCR. Cage PCR is very good and very sensitive in general at finding pinworm at the cage level. So all the fecal floats that we did in the autoclave rooms and the non-autoclave rooms that were positive on plenum PCR were negative. All the pool cage PCR swabs were also negative. So we have to conclude that really the exhaust plenum PCR do not correlate well with fecal flotation or cage PCR results from index high-risk colony animals. We also said, well, you know what, the probability that we have a selective outbreak in non-autoclave rooms is really not very, does not hold too much. The second thing that we said, well, maybe we had historical presence of pinworm DNA in our exhaust manifold without any active infection in our colony animals. However, several of our racks that were positive on exhaust plenum PCR had been fully decon and autoclaved just 1 or 2 months prior, and the autoclaving should have denatured the DNA. So that suggested that really the contamination would have occurred after those dates. The historical contamination would not have explained also the discrepancy between the autoclave and non-autoclave room. They are processed the same way and they're circulated through the different rooms the same way. We don't dedicate racks to autoclave rooms and dedicate racks to non-autoclave room. Once they're clean and autoclave, we circulate them back into different rooms. So we just put that hypothesis aside. I have to say another thing is that in our exhaust plenum, we found some Booklets, and just want to touch base really quickly. Sorry. We found some Booklets in our plenums, and we said, well, could that be a source of false positive due to cross-reaction? Because maybe it has a very similar genetic material. Well, in fact, it doesn't, but we tested it anyway. We tested those Booklets by PCR, and they were negative. Good. And second thing that excluded that possibility is that both autoclave and non-autoclave racks in both autoclave and non-autoclave rooms had Booklets. So it probably didn't come from the Booklets. So looking back at the data, we want to say, how can we explain the discrepancy between the autoclave and non-autoclave rooms? How could we explain the widespread positive results on plenum PCR affecting selectively non-autoclave rooms? Well, it had to come from a component, the source of the contamination had to come from a component used inside the cage throughout the facility that is neither sanitized nor autoclaved in the non-autoclave room. So only 2 components met these criteria, either the corncob bedding or the nesting material. So the nesting material, the crinkle nest, was not our first suspect because it's a paper product. It doesn't have any nutritional value and it's unlikely to draw infected vermin. Nonetheless, we excluded the hypothesis by just testing by environmental PCR all the packaging material for the crinkle nest that we had in our facility, the room where it was stored and unpacked, and the room where it was distributed from. All the results were negative. Although it doesn't prove anything, the probability that it came from the crinkle nest was fairly low. Corncob at that time was our most probable source. We said, you know, it's an organic material, maybe it was contaminated, and we didn't know a lot about corncob. Well, we know what we knew based on what we had read, but that's about it. So we swapped the automatic bedding dispensing unit for our corncob. All our corncob is dispensed into cages by a centralized automatic bedding dispensing unit. And so we said that's the best place to test our corncob, right? So we tested that for pinworm for pinworm by PCR. And to our great surprise, it was positive. And I said, oh my gosh, our automatic, either the corncob is infected, or the bedding dispensing unit has vermin in it or is infected. So we emptied the reservoir and inspected for pests, but we found none. We also did swabs everywhere in our storage area. It was all negative. We also did swabs of at our corncob distributor, in their storage room, of their delivery trucks. All the results were negative. So we started to say, well, maybe the contamination occurred earlier between the manufacturing plant and the distributor. And we said that would be logical. Basically, you have a contamination of the corncob somehow after the production, and it came into our facility, and that would explain why it spread throughout the facility, because it's distributed through our centralized bedding system unit. But that didn't explain the discrepancy between the exhaust plenums, the sentinels, and the colony animals. Except in the 2 rooms where we did confirm the positive pinworm, the pinworms, none of the exhaust plenum PCR results were confirmed in sentinels or colony animals. So we started to doubt the specificity of the PCR assay that we've used for our plenum PCR. So to test that, we said, you know what? We're going to take the same plenum swabs from the 2 confirmed positive rooms, from non-autoclave rooms that were positive by PCR and by autoclave rooms that were negative. And we submitted those samples to 2 labs, saying maybe they have 2 different primers, 2 different assays, and if one is not specific, maybe the second one is specific, and then you're going to have a negative result. So the samples were submitted to the original reference lab and to a second reference lab. And big surprise. As expected, well, the first reference lab in all non-autoclaved rooms were positive. Again, the plenum PCR were positive when tested by the first lab in non-autoclaved rooms and negative in autoclaved rooms. That confirms exactly the results that we had before. But when we got the results from the second reference lab, what we had was that all the results were negative in non-autoclave and autoclave rooms. Very interestingly is that in non-autoclave rooms with confirmed PCR, even in those rooms where we did confirm pinworms, the plenum were negative. So our conclusion was very simple, that the primer used by the original reference lab was not specific and just amplified another nematode. And the second thing that is as important is that the PCR of exhaust plenum is not really efficient in detecting pinworm in naturally infected mouse colony. Yes, indeed, staph 44, staph 50 in red in the table were confirmed positive in the animals, and yet the plenums were negative. So to test the specificity of the primers from the first reference lab, what we did is we sequenced the amplicons that were amplified by that PCR reaction from the exhaust plenum samples. So we sequenced the amplicons, and then we blasted that sequence on GeneBank, which is a genome database. And what we found is a 100% match for 3 nematodes of the Rhabditid family, completely different from pinworms, suggesting that the primer non-specifically amplified DNA from other nematodes. We then compared the obtained amplicon Rhabditid sequence with the known sequence of Aspicularis and found a 91% sequence identity with the Aspicularis 18S ribosomal RNA gene and 100% match in the region of the primer. So obviously, with those results, we concluded that the Aspicularis positive results on our exhaust plenum by our first reference lab in the non-autoclave room was a false positive due to the non-specificity of our primers. So I just want to touch base on nematodes in general. The nematode phylum is very rich in species, and it's very biologically diverse. You know, nematodes infect plants, animals, and a lot of them are free-living organisms that we find in soil, etc. We estimate that there are between 1 and 10 million existing nematodes, 1 to 10 million existing types of nematodes species, but only 25,000 are actually described in the literature, and only a few have been sequenced. As far as the Rhabditid nematodes, what are they? They are the richest group of nematodes. They have about thousands of members, many which are undiscovered. They're free-living saprophagous parasites present in every terrestrial habitat, but especially in soil and sediment, because they feed on decaying matter, organic matter, including plants and bacteria, and they just convert that, they just destroy that organic matter. So they have a very important ecological function. They are extremely, extremely resistant in the environment. I'm not talking about resistance that, when you're talking about resistance of pinworm, that's one thing. But those bugs, those Rhabditid nematodes, I'm talking you're finding them in remote caves that under so harsh condition of pH, temperature, etc., and they still are resistant. So, and that's very important because you have now a Rhabditid nematode that is ubiquitous in nature and that we found in organic product, namely Corncob. So what we said, it was probably a post-production contamination of the Corncob bedding with that nematode, or just genetic material that remain in there, and that caused the false positive results in our plenum PCR. And that would make sense. It would make sense because it was a widespread outbreak, well, a widespread dissemination of that nematode in all our non-autoclave rooms, and it probably came from the corncob. But I just want to touch base on corncob bedding because I think it's important to understand. I hear a lot of things, and I'm the first one to sometimes draw conclusions too fast. I've heard a lot of things. Well, corncob bedding can be infected with mouse pathogens. Well, we've never had confirmed infection of our animals with mouse pathogens from the corncob. And when you look, when you hear all those things, you better go back to the source and try to understand how really this is, how really the corncob is done, to realize that it's almost impossible that contamination of the corn before production could pass through the process and then be distributed to your facility. It's almost impossible. I took an airplane ticket and I went to the Midwest and I decided to go to a corncob bedding, corncob manufacturing plant to try to understand if that was possible. So the corncob is made from the central core of the maize plant, not the outside, just really the core, and it's passed through different hammer and roller mills to produce fractions, and then it's heated dry to over 200 Fahrenheit, and that's really high. And then the sizes, the fractions are sized and separated by sifting, and they're dust aspirated. Basically, corncob bedding is produced under extremely harsh condition, and I don't see anything that could contaminate the corn before production and that could survive that process. I don't believe it. And the second thing is that the corncob that I saw at QA programs, everything was done under strict quality control. It was done under great conditions. Everything was controlled. Now, that something might have happened and that the corncob, in our case, got contaminated with soil or something like that after production, in between the transfer, after production and during transfer or whatsoever, that's possible, and that's probably what happened, because the nematodes that we found are ubiquitous in the nature. But that something infectious might have passed through that process is almost impossible. So what have we learned from this? Well, first we need to remain critical whenever we use a diagnostic methodology. Don't take results for granted. Ask questions. Use your common sense and your intuition before implementing very costly and disturbing containment and eradication plan. Choose a diagnostic lab, and I think that's essential, that will work with you and go the extra mile to answer your question. All that what you've seen there, we did in collaboration with our reference lab. In this case, we have developed an amazing relationship with our reference lab. That their primers were not specific did not matter. Why? Well, simply for a very simple reason. We use an assay that they validated to test biological samples in a mouse. And we experimented. We played the apprentice sorceress to test environmental samples. This was purely an off-label application of their PCR assay. Their test was never validated for environmental samples, and the primers could not have been validated to account for the thousands, if not millions, of nematodes in the environment that could have cross-reacted. However, think very closely. Would your reference lab have worked as hard and close as ours to get to the bottom of all this? Ours didn't, and that's why we never considered changing. They understand that diagnostic testing is not an exact science. And that's what we value in a diagnostic test, in a reference lab. Like any other diagnostic test, PCR is subject to false positive and false negative. Any results must be interpreted cautiously with an overall understanding on the methodology and all the intrinsic controls and all the variables that may affect the results. Many aspects of the methodology should be considered, including but not limited to the primer design and the specificity of the primers. Any potential source of contamination of the sample that could occur during manipulation at your own facility, but also anywhere across during the transport and at the diagnostic lab. You also should understand what type of PCR assay do they do. Did they do a simple PCR, a quantitative PCR, a multiplex qPCR, or a nested PCR, which is meant to reduce the non-specificity binding by using 2 sets of primers. You need to understand all of this before you finalize your analysis and say, well, that's a true positive. And that's only a short list of things you need to understand. The second thing, just to give you an idea, if you did a search on the validation of diagnostic PCR assays, the validation of PCR assays in general in human and veterinary medicine is very challenging. If I just did that little exercise, I did a PubMed or Medline search and looked at all the publications that were done to validate a diagnostic PCR assay for TB in humans. TB is very common. You find about 70 to 80 scientific articles in peer-reviewed journals just for the validation of that assay, under different conditions, with different population, in different countries, etc., on different samples. If you did the same search on the validation of a diagnostic PCR assay for MPV, mouse parvovirus, in mice, you would find about 2 or 3 articles. That's about it. So an amazing amount of work is done to validate those PCR assays, but you should not think that they are bulletproof and that, especially in our small field, everything is done and redone by multiple teams and that they are completely bulletproof like they might do it in human medicine. And even in human medicine where they have so much resources, well, some of their PCR assays do fail. Not to say that we don't do enough work or that the validation process is less stringent in lab animal medicine. Absolutely not. But again, we should not think that they are bulletproof and that they're going to work. So how does the validation of those primers or those PCR work? Well, first, the validation starts with the design of the primers by computer modeling to optimize the PCR condition. We take into consideration primer length. In general, the primers are about 18 to 22 base pair. Primer melting temperature, primer annealing temperature, GC content, and any secondary structure like hairpins, self-dimers, or cross-dimer that may prevent the reaction. You know what? If you have very loose PCR reaction, you can anneal a primer to almost any DNA sequence. You got to design those primers very specifically and very with that computer model to make sure that when it anneals to your sequence, it anneals correctly and very strongly. Second thing is that the primer needs to be designed to be specific. That's a given. So what normally reference labs do is they normally target ubiquitous housekeeping genes or ribosomal RNA common to several species to identify the pathogen at the genus level. Then one or a few sets of primers, depending on the type of PCR, are designed to target specific elements for organism speciation. Then what we do is the specificity of the primers are tested and compared with all available genome databases. And I want to specify, I want to emphasize that all available. For primer design for biological samples in a mouse, it's very simple. We know, that pinworms, what infects the mouse. That's only a few species. Good. But when you're looking at the environment, we're talking about millions of different nematodes that might cross-react, and many of them that have not been sequenced yet. Then the in vitro sensitivity of the primer is verified by using tenfold dilutions of target pathogens. Then the specificity is evaluated using DNA extracted from large banks of pathogens of the same family. And then the in vivo specificity and sensitivity of this primer is validated by testing known clinical samples in the presence of positive and negative control. So that's how normally the primers are designed. So validation of PCR assay is a very stringent one that might give you the false impression that it's bulletproof, right? Well, don't fall under the illusion. It's almost bulletproof. Right? Don't get me wrong. It's 99.9% bulletproof, but it's not, and it can fail. In our case, the PCR assay from our first reference lab targeted a region of the 28S ribosomal RNA gene common to Cephalosporin muris, Velata, and Aspergillus tetraptera. It was validated according to the same stringent process, and but the PCR assay was not validated to test environmental sample, and so it's not surprised that we had non-specific results. And why is that? Well, first because genome database are continuously evolving, and basically they are flawed, incomplete, and sometimes inaccurate. Most of it is accurate, but you do have some flaws in it. Many of the genome database contain unverified or incomplete or inaccurate sequences. And they also contain entries from genetically different organisms under the same species, as well as and every year there's a large number of microorganisms that are discovered every year and that are yet to be classified or sequenced. Again, nematode phylum is very broad. You have about 1 to 10 million nematodes in the environment that could cross-react when you do environmental PCR, as opposed to PCR on biological sample. So I hope that I have convinced you that although the validation process of a PCR assay designed to test a mouse-specific pathogen in a biological sample from the animal might look straightforward, it's absolutely not true for environmental PCR. There's so much more uncertainties involved, and so many other possible source of false positive results. I just want to touch base on again and come back on plenum exhaust plenum PCR. What is out there in the literature? Because remember that statement at the beginning in the publication that we could replace soil bedding testing by exhaust plenum PCR. Well, let's go look at the literature. What do we have? What they found is that plenum PCR was very good for MHV and Sendai to monitor those bugs or those viruses at the rack level in experimentally infected mice. That's very important because experimentally, when you do experimental infection, you give them a huge amount of virus, and their shedding is much higher than in natural infection. So basically, you optimize your chances of finding it at the exhaust plenum, but doesn't represent what happens in real life. The only one publication that I've seen that seems to show that exhaust plenum can be useful to assess colony health status was for firmites. That was in a natural infection. And also I saw some on certain bacteria like Corynebacterium. But the results from exhaust plenum to monitor colonies infected for MPV or Helicobacter, that's much more variable. And as we've shown in our case, to monitor a colony that is positive for Aspicularis tetraptera is not good. Remember, our 2 rooms that were confirmed positive were negative by the second reference lab on plenum PCR. Overall, what we need to understand is that there's no systematic study that have validated environmental PCR from exhaust plenum for different organisms in natural infection, or that have determined the optimal sampling strategy to monitor colonies. When to sample them, where to sample them, how often to sample them. There's just no data out there. So remember that statement we found in the publication? Well, although the technology of plenum PCR is very, very promising, we are just not there yet. Not to say that there's no need for it or that it cannot be used as a complement to soil bedding sentinels. Absolutely not. But we need much more studies and more work to determine a safe and efficient way to use it. So what have we learned from all this? Well, PCR can yield false positive results despite all the safeguards used by labs to ensure that the specificity of their assay. Environmental PCR bears unique inherent risk that we need to take into consideration. Always ask questions and confirm unexpected positive result by a secondary method. That's essential before implementing costly and containment and eradication strategy. If we had trusted our plenum PCR and didn't ask any question, believe me, we would have treated everybody in our facilities, and it would have cost a huge amount of money, and it would have caused a huge headache to a lot of our investigators and ourselves. Another conclusion is that there's no systematic study that have validated environmental PCR from exhaust plenum for all the different microorganisms seen in mice. And there's no study that have determined the optimal sampling strategy. And finally, the environmental PCR from exhaust plenum is not efficient, in our case, it was not efficient in detecting a Spicularis tetraptera in natural infected mouse colony. So I think that's about it. I'm sorry I took a lot of time. If you have any questions, please go ahead. I'll give you control. I'll give full control. Pass full control. Judy, do you have the full control? Control?
Yeah. Thank you for that informative presentation. Before we get started on the question and answer session, I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. And our first question is from Judith Nielsen, University of North Carolina. Do you irradiate or autoclave the bedding on most all cages?
Sorry, could you repeat the question because I didn't hear it?
Okay, the question is again from Judith Nielsen, University of North Carolina. She asks, do you irradiate or autoclave the bedding on most all cages?
Wait a second, I'm going to get this. The answer is no, we don't irradiate or autoclave. Basically, the corncob bedding is the vast majority of our cages are not autoclaved. Only 5% of our cages are autoclaved with the bedding and the crinkle nest. And we don't irradiate the corncob.
Okay, our next question, I apologize if I mispronounce your name. Our next question is from Cleda Silva, Fundação Hemocentro de Ribeira Preto. How do you confirm the parasitism extension?
So if I understand well, how do I confirm the parasite extension? I'm not sure I understand the question per se. If you mean that how did we confirm that it, well, we had environmental results, that's one thing, and then we needed to confirm in the animals. So did it extend so was there a correlation between our PCR results from the plenums and in the animals? And the answer was no, there was not. So if that's the question on how we confirm the extent, that's how we confirmed it. We also confirmed the extent in our facility by retesting our sentinels and by testing index high-risk animals in all the rooms. So that's how we did it.
Okay, our next question is from Maya Meeks at Emory University. She asks, are your facility Sentinel program SOPs available online?
No, they're not. It's something that we keep. But you know what? Those SOPs are fairly standards. The soil bedding sentinels are. It's fairly standardized. The way that we pass on it's what you will see in the literature. There's a lot of good articles. I think there's an article that was published very recently on soil bedding sentinels, based on PHILASA and ALAS. But what we do is fairly standard, and we don't put it on our website, or we don't make them publicly available. I'm sorry.
Here you have another question from Judith Nielsen, University of North Carolina. How did you collect the samples for pool cage PCR testing?
So that's a good question. Basically, for the pool cage PCR, at the cage level, what they've shown is that you put a swab, and there's an SOP on Charles River as well as Radel for that. For those techniques, you just put the swab at the junction of where the corncob level is in the cage, and you go all around the cage, and you go around the cage and swab at that level, at the level of the cage. That's for the environmental cage PCR. And the pool, I think you cannot pool more than 10 cages per swab.
Okay, Aurore Daudet-Devier from the Université de Montréal asks, at the manufacturing level, is the corncob ever tested for rodent pathogens?
To my knowledge, at the manufacturing plant, the answer would be no. I don't believe that they test for mouse pathogen at the manufacturing plant. Now, I just want to emphasize something. When you go there and see what they do and how they do it, if you had a contamination at the plant level, because those corn are taken from cornfields, they're brought into and they're put on in huge mountains, and then they're processed through the plant. The production of the corncob is so harsh. The temperature is so high. Everything is, the production conditions are so high. I don't see any mouse pathogens that could survive. If there is a contamination that is possible, it's post-production, not, it wouldn't be pre-production. I'm fairly convinced. And having looked at it and spent 2 days over there and looked how they do things, I'm convinced that that's almost impossible. So, however, there could be a post-production contamination. But that's with the food, that's with any products that is produced. It could be anything from during transport. It could be after the bagging, transport. It could be while you unpack. It could be anywhere. But again, that you have a pathogen that goes from before the production that infects the corn and then passes through the production or the processing of the corncob. I don't believe that's possible.
Okay, we have time for one more question, also from this same questioner, Aurore Daudet-Devier. How do you know if the PCR is validated for environmental use?
Well, that's the problem, is that they don't validate it for environmental use. Most of the PCR that you have out there are validated for use in biological samples from the animals, not for environmental use. And you can see, based on what I've tried to explain, is that validating an environmental PCR is much more difficult, because you just don't, you're faced with so many other types of organisms that can cross-react. Now, don't get me wrong. We were very unlucky. We were very unlucky because the probability that, the PCR, our environmental PCR cross-reacted and the primer was not specific was very low. I'm not going to say that it's a huge risk, but you cannot say that those PCR, especially for environmental PCR, are bulletproof. There's no validation that will allow that. However, they do give us an interesting tool to assess environmental samples. But overall, what needs to be done is, whenever you have a PCR result, you need to confirm in your animals. You cannot sit and say you have an environmental PCR that is positive and say, we're going to take that as a positive. You need to confirm in the animals. That's your gold standard. If you have it in your animals, then it's true. However, if you have a positive on environmental PCR, it might just mean that you have historical contamination or DNA present in the plenum, but no infectious particles in the animals. It could mean that you have a false positive results. It could mean a lot of things. Always come back to the animals. Always confirm in the animals.
Unfortunately, we are out of time. I want to thank the audience for their interesting questions and participation in today's event. I would also like to thank our sponsor, the Anderson's Lab Bedding Products, for making today's educational webcast possible. Today's webcast will be available for on-demand viewing through August 2015. You'll receive an email from Lab Roots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. See you next time. Goodbye.